Okay, I think I'll just get started now. Did everyone sign in? I'll give you a second if you haven't. Oh, I also wanted to add this semester, if you ever can't make it to a session or are watching it online, because I do post them, if you can sign in, that's helpful too, so they know that SI is being used. Okay. So the first unit is blood. I know you guys did this last week, and now you're on heart, if I'm right. Okay, yeah. So then this week you're doing heart part one, and then next week is heart part two. So this SI I focus mainly on blood, and then next week I do both of the heart units together. Yeah, and I love these units, so super exciting. The blood and heart ones are really cool. Okay, so office hours now are Fridays from 2:30 to 3:30 in the Academic Achievement Center. So definitely come, ask me any questions you have. Or you can email me if you can't come to that. And my notes and slides are available on Husky CT. Something to note there is I took this class 2 years ago, I think. So my notes are broken down by blood and heart part one was our first exam. So if heart part two isn't on that same note sheet, just go and look at the next note sheet and you'll find it. So different organization, but it's all there. Okay, starting out with some blood characteristics. It's a fluid connective tissue, and it's a homogeneous mixture, which just means it's made up of a bunch of things, but it looks like it's just one thing. Yeah. Some general properties. It's mostly water, as we'll see in the breakdown in the plasma. It has a neutral pH of 7.4. Volume of 4 to 6 liters, which is 7% of body weight. And then men have 2 liters more because blood volume is proportionate to body size. Just to note, I'm very general right now, but I will get more specific as we go on. So here are some main functions of blood transport. So as you can imagine, it's taking all of the nutrients, gases, wastes all throughout your body to different organs to be processed in tissues. Like we said, it has a pH of 7.4, so that's providing an optimal environment for all of your cells and body processes to function correctly, the right pH for proteins and everything. It maintains a body temperature, 38 degrees Celsius. Since blood is mostly water, it kind of helps maintain the body's temperature at that value because water has a high specific heat. So that's a good temperature buffer to maintain homeostasis inside the body. It also regulates osmolarity by having different concentration of solutes, and it maintains osmotic balance in the tissues. And it has this property called hemostasis, which prevents fluid loss. So it's basically blood clotting. So it protects your body from losing blood and staying together. And then it also protects you by being a major component of the immune system, as we'll see with white blood cells. So here's the main breakdown of components of blood. So there's the plasma, which is mostly water, but also has some important proteins, and it includes the gases. And then the formed elements are leukocytes, which are the white blood cells. And you get more into that in lab, the different types of leukocytes, like the granulocytes, agranulocytes. Not as important in lecture, but there's the breakdown of those two categories at the bottom of the screen. Then you have platelets, which are involved in hemostasis, and erythrocytes, which are the red blood cells. So here's that broken down with some more details. So the plasma is 92% water. That's a lot of water. The plasma is also known as the supernatant. And in total, it's 46 to 63% of blood. So then 7% is plasma proteins. A big chunk of that is albumin, which I'll talk about in the next slide. Then you also have globulins, which transport the antibodies and other globulins. Fibrinogen, which is essential for blood clotting, helps with hemostasis. And then some other regulatory proteins like enzymes. And then 1% is some other solutes like electrolytes, organic nutrients, and organic wastes. So that's like sodium. You can remember that's important for neurotransmission, like depolarization, repolarization. Organic nutrients, lipids, carbs, amino acids that like make up your cells, and also like indigestion. And then organic wastes, urea, creatine, and bilirubin. So for the formed elements, these are also known as the pellet. This is 37 to 54% of blood. And red blood cells are like almost all of it, 99.9%. So we'll talk about those soon. So the majority of the plasma proteins in the plasma are albumin. They make up 60% of the plasma proteins. So a little bit about this one is that it functions in maintaining osmotic pressure in your body. So you can like alter an environment by increasing levels of albumin. It then like water kind of comes with albumin because it can't leave the blood. So if you increase levels of albumin in the blood, you're gonna then like increase the volume of blood by like drawing more water into that area. So it can minimize water leaving the blood by increasing osmosis, increasing water retention in the blood by increasing levels of albumin. Yeah. Any questions so far on anything? We're doing good. Good? Okay. So now moving on to the formed elements, 99.9% are the erythrocytes. So the main function is to deliver oxygen to your tissues and remove carbon dioxide. So the morphology is really important. They're biconcave, which means they have like that thin center and are thicker around the edges, kind of like a donut shape. So that just increases the surface area and allows them to like slide nicely through the small capillaries, your blood vessels. So that also makes them stackable and flexible, so they can, again, like go through the blood vessels without like getting stuck. So the way that red blood cells can transport these gases throughout the body is because they contain hemoglobin, which is a protein that is responsible for the gas transport. So each hemoglobin contains 4 molecules of iron, and each molecule of iron serves as a binding site for oxygen. So that's why each hemoglobin can transport 4 molecules of oxygen. Hemoglobin is also the most abundant protein in the red blood cell. And hemoglobin itself is a spherical globular protein made of 4 subunits, or globulins. So there are 2 different types of hemoglobin. When you're an adult, you have a certain type, and then when you're a fetus, you have a different type. So in adults, you have 2 alpha globulins and 2 beta globulins, as you can see, like the yellow one on the bottom right. And then fetal has 2 alpha and 2 gamma. So what that basically does is having the different globulins changes the hemoglobin's overall affinity for oxygen. Yeah.
I think you mean globin.
Oh, globin. Okay. Sorry. Oh, yeah. Globin. Okay. So, fetal hemoglobin. Oh, you mean, wait, what? Just like the way I'm pronouncing it?
Yeah, because globulins are something else.
Okay, yeah. Okay. Thank you. So, yeah, just by having the different globins for fetal hemoglobin, you can then increase its affinity for oxygen. So when you're a fetus receiving like oxygen from an indirect source, the mother, it's easier for the fetal hemoglobin to then grab onto that oxygen. Yeah. Okay. So a major kind of property of the red blood cells is cooperativity. Um, so basically, the hemoglobin changes morphology when the gas binds to it. So when one molecule of oxygen binds to the hemoglobin, it changes the structure of the other chains and allows more oxygen to bind to it. So it's not like a linear graph, you could say. Like if one molecule of oxygen binds, then the other molecules of oxygen are going to bind easier, which is what cooperative binding is. So then there's different cooperativity of the heme groups. So there's the Bohr effect, which means that binding of carbon dioxide decreases the affinity for oxygen. So it's the opposite of when oxygen increases the affinity for itself, carbon dioxide decreases the affinity for oxygen, which we'll talk about with the dissociation curve. This leads to a right shift. And then there's also the Haldane effect, which means that the release of O2 increases the affinity for CO2. So when the hemoglobin releases oxygen in the tissues, it then can more easily bind carbon dioxide, is what that means. Okay, so here's the hemoglobin oxygen dissociation curve. So it describes how oxygen and hemoglobin interact, which basically means that it says that the amount of oxygen bound to hemoglobin depends on the amount of oxygen in the environment or the plasma, which is the partial pressure of oxygen on the bottom. So the x-axis on the bottom is basically just telling you how much oxygen you have available in your blood, in the plasma. On the y-axis, you have the hemoglobin saturation. So P50 is looking for 50% saturation, or in other words, when half of the iron groups are bound to oxygen. So 2 out of the 4 iron groups would have an oxygen bound at P50. I know that this one can be a little bit hard to understand, so does anyone have any questions about the curve so far? Okay. So now you got the right and the left shifts. So different environments, conditions in the body, or like we were talking about, fetal versus adult hemoglobin can change the hemoglobin oxygen dissociation curve. So if you have a right shift, meaning that line is shifted physically to the right, then that means you need more oxygen. You need more oxygen in the plasma to reach the same P50 or 2 oxygen bound to the 4 iron. So that, in other words, that means you have a decreased binding affinity. because you need more oxygen in the environment to bind the same amount to the protein. So then here are some things that cause the right shift. Carbon dioxide, like we talked about before, this is the Bohr effect. So if you bind CO2, that decreases the affinity for oxygen, and therefore you need more oxygen in the environment to reach the same amount of oxygen bound in the protein. Acidic conditions also lead to a right shift. DPG, which is just a byproduct of the body. Exercise. So that makes sense. Like if you're exercising, you need to take in more oxygen to get the same amount of oxygen bound to your hemoglobin. And a temperature increase. So another way to think of the right shift is right shift release. It's not holding on to the hemoglobin. You need more oxygen in the plasma to get the same amount of oxygen bound to the hemoglobin. Left shift is the opposite. So you need less oxygen to reach the same amount bound to the hemoglobin. So in other words, you're increasing the binding affinity for oxygen here. You need less oxygen in the environment to still get the same amount bound to hemoglobin. So some things that cause a left shift are fetal hemoglobin. So like we were talking about before, the fetus needs to really grab onto that oxygen because it's getting it indirectly from the mother. So you're going to have an increased binding affinity. Inactivity. So you can think when you're like sitting down and resting, you're not like breathing really heavily. You're kind of taking in less oxygen than you would be exercising. So on that x-axis, you're going to be closer to 0. So you need less oxygen to still have all of your hemoglobin having 2 oxygen bound. Cold temperatures, and then also basic environments. Okay, so I was going to try to draw it so you guys could like better interact with it maybe. So again, on the x-axis, we have the partial pressure of oxygen. So this is just that's just the amount of oxygen in the plasma. And then on the y, we have the hemoglobin saturation. So at 50, this is what we call P50. Which is 2 molecules of oxygen bound. Okay. And then you get something that looks like that. And normally to find P50, you would just go over to 50 on the hemoglobin saturation, and then draw a line here, draw a line down here, and then whatever the partial pressure of oxygen is in the plasma would be the P50. So let's just say it's like 30. So in this case, it would be 30. And then a right shift would physically just mean that you're shifting the line to the right. So when analyzing this graph, you can tell that when you shift to the right, you are then going to need more oxygen in the plasma to reach the same amount of saturation. So right shift means decreased affinity. On the other side, if you have a left shift, then you need less oxygen to reach the same level of saturation. So you have an increased affinity. You're grabbing onto the oxygen easier. So there's like the same thing. Okay, so we've talked about this a little bit, but fetal hemoglobin has an increased affinity because it gets it indirectly from the mother. It needs to grab onto it if it can. So would fetal hemoglobin show a shift to the right or left on the hemoglobin oxygen dissociation curve? Yes. Is that clear to everyone, like why? Yeah. So if you look here at the environment, if the fetal hemoglobin has an increased affinity, it doesn't need as much oxygen to reach the same amount of saturation, basically. So there's basically, it has like, yeah, it needs like less oxygen to grab onto it. Like if you think of it as kind of like a pool with like a bunch of oxygen in it, the fetal hemoglobin is so like good at grabbing it that it needs like less oxygen in the pool than one that has a harder time grabbing it would then need a lot more to pull from, basically. Yeah, so then over here, so that's the same as what we have. Moving on to the next formed element, the leukocytes. These are the white blood cells. They're about 0.1% in combination with the platelets. These provide defense against infection, and they are very diverse. There are 5 categories. And you'll, like I said, you'll get more into these in lab. I don't think you have to worry about them too much for a lecture. And then the last formed element is the thrombocytes. So these are fragments of megakaryocytes, and they're critical for clotting or hemostasis. So, although these are just 0.1% in combination with the white blood cells, there's a little bit more of them than white blood cells, which is why they're intermediate abundance. The number of platelets are regulated homeostatically. So then there are these two conditions where you have disturbed platelet regulation. You have thrombocytopenia, which is when you have less than 80,000 platelets. So you don't have enough. And so if platelets do blood clotting, you then are going to have issues with blood clotting. You're probably going to lose a lot of blood if you get a cut or something. Then you have thrombocytosis, which is more than 1 million platelets, and you have too many, so you get too many blood clots. And then some factors that make platelets good clotting factors is that they release pro-clotting factors, they aggregate, and they're contractile.
Should we know the exact number of platelets, or does it not roughly?
I would just know that thrombocytopenia is not enough. And then the effect that's you can't have blood clotting, and then thrombocytosis is too many, so you have a predisposition to clot. So we've talked about this, hemostasis, blood clotting. Here are some clinical tests to analyze blood. You have the hematocrit, which is the percent of total blood volume that's red blood cells. This is a very simple, quick, and easy test. And then you have complete blood count. And this is just kind of like getting a little sample in a microscope and counting them to estimate the proportions of blood that are like red blood cells, white blood cells, platelets. And then the differential is a closer look to identify the subpopulations of white blood cells. So like we talked about before, like the granulocytes, neutrophils, stuff like that. And that could help you identify very specific diseases. So this is how the hematocrit's done. So you put it in a tube, and then you throw it in a centrifuge, and then the blood fractions into plasma, white blood cells, and platelets, which is also known as the Buffy coat, and then the red blood cells. And the hematocrit itself is just like the percentage that is red blood cells. And it's normally about 40%. And then here's a quick look at the CBC and differential. And what like the different acronyms stand for. So white blood cell, red blood cell, HCT is hematocrit, HGB is hemoglobin, and then MCV, mean corpuscular volume. And then that's just the average size or volume of red blood cells in the sample. So it's telling you if your red blood cells are too big or too small or normal size. This is some information on blood typing. So you have two classification systems, ABO and the RH system. So the ABO system refers to the antigens on the red blood cell. And then if you have a specific type of blood, like say you have type A blood, you then have A antigens, and you have the opposite type of antibodies. And your body is regulated that way because you don't want to have the same antigens as antibodies, because then they'll bind and clot. So then the different ones are type AB has both A and B antigens on its surface, and then you have no antibodies. And type O has no antigens, and therefore you have A and B antibodies. So here's what that looks like. You can see type A has the pink antigens on the surface, and then B antibodies in the plasma. And then this is the same thing, but taking into account the RH factor. So if you have the RH factor, you're RH positive, so it's just positive. And if you don't, like at the bottom, you're negative. So specific blood types can only accept specific blood types because of the reactions between incompatible antibodies and antigens. So if you get the same antibody and antigen, you're gonna have agglutination, which is just like clumping. So antibodies are removed from the donated blood because the plasma isn't donated, just the red blood cells are donated. Okay, so using this logic, what type of blood can type A blood receive? Does anyone know?
Type
What'd you say?
of antibody.
Yeah, it can receive A and then it can receive one more.
Oh,
Yeah. And then B, yeah. A, B, A, B, O. It can receive some more than that. Yeah. Perfect. And then O, yeah. Cool. So if you're Rh positive, you can receive Rh positive or negative. A person who is negative can only receive negative. All right. Okay. So this is a hemolytic assay. You guys did this in lab last week? Okay, cool. So basically what you're doing here is you're adding the antibody to each one to see the reaction, to see what clumps. And if you get clumping, you can therefore infer what type of blood type you have. So does anyone know what the first one is?
A positive.
Yes. Is anyone confused as to why it's A positive?
is it because the agglutination?
Yeah. So since you're adding antibody A and it clumps with the A, that means that you have type A. And then since you get clumping when you add the anti-D antibody, you then are positive for the Rh factor. Does anyone know what the next one might be?
B positive.
Yeah. You guys seem pretty good on blood typing. I feel like this is, people usually struggle with this. Next one,
AB positive.
yeah. And then the last one,
Oh,
O negative. Okay, so I don't know if we're gonna try this. So I have some questions on Top Hat. If you guys want to go to Top Hat. Can you guys see that? You can?
yeah.
Okay. So that's what type of blood types have B antibodies? Oh, cool. Okay, I feel like answer D should not be there. Because it would be, you guys got it. Type A would have B antibodies, but then also type A plus would have B antibodies. So B is the correct answer. Does anyone know why type AB would not have B antibodies?
Because it has B antigens, so it would
Yes. What type of blood is known as the universal donor? Can you guys see this?
Yeah.
Okay. So universal donor meaning that this person could give blood to any other blood type. Did you guys put an answer? You did? Oh, okay.
I think because you put it as review and not as homework.
Oh.
You can't see it.
Okay. I'm not gonna do that. Okay, well, I'll just do a better next one. Do you guys say type O? Okay, cool. Universal acceptor. Yeah,
Is there a universal receiver?
that's this one.
I don't know.
Nice. AB. Does anyone want to explain why?
Because it doesn't have any antibodies.
Yeah. So AB doesn't have any antibodies, so we could take any type of blood with any antigen. Fetal hemoglobin has a blank affinity for oxygen, causing it to display a blank on the curve. Perfect. I feel like I drilled that, so I feel like that'll definitely be on the exam, I'd expect. Which is correct about the classification of blood? Okay, yes. So I was trying to be a little bit tricky with D, that it's about 90% water. So this one's just like read carefully because the plasma is 90% water. Overall, blood is like 50% plasma, 50% formed elements. Of the plasma, it's 90% water. It's not only plasma, and then it's not heterogeneous, it's a homogeneous mixture. Okay, so this is kind of like I do have the picture on the board, but if it wasn't there, I mean, maybe it is a confusing question. Okay. We're pretty split. Does Dr. Redden usually have it so you can see like what people are putting as you're responding? Okay. Okay, so for this one, I was thinking it's found on the x-axis because you have the hemoglobin saturation, but then you're going over and looking and finding the actual partial pressure of oxygen on the x-axis. So you're finding it here on the x-axis. This is if they ask what's P50, it would be 30 for the middle one. A left-shifted hemoglobin oxygen dissociation curve. What? Yeah, so nice. First C, that one would then be a right shift. If you have a decreased affinity for oxygen, you need a higher percentage of oxygen in the plasma to reach the same amount of saturation. Did you guys like these questions better than the PowerPoint? Yeah, okay, so I'll keep doing this. Does anyone have any questions about blood that they want to talk about or go over? Anything confusing? Okay. So this is a reminder also, like I don't do everything that you need to know for every unit. So here are some things I didn't get very specific into. Hemostasis, the red blood cell life cycle, and then all those like blood conditions. If you have questions on those, you can like look at my SI notes that I linked there, or come to office hours or email me. And then that's all I have. So thanks for coming.